Not all neurotransmitter receptors are ion channels. Many are coupled to enzymes that produce intracellular second messengers, chemical signals generated inside the cell that then go on to regulate ion channels and a wide array of other proteins. This is how neurons can have prolonged modulatory responses to neurotransmitters rather than just rapid on-off switching. One major pathway begins when norepinephrine binds to beta-adrenergic receptors. This activates the enzyme adenylyl cyclase, which converts ATP into cyclic AMP, abbreviated as CAMP. CAMP then activates a protein kinase called CAMP-dependent protein kinase, which phosphorylates voltage-gated calcium channels. Phosphorylation means adding a phosphate group to the channel protein, which changes how the channel behaves. In many cases, this increases the duration of time the channel remains open once activated, thereby enhancing calcium influx into the cell. This pathway illustrates a key principle. A neurotransmitter binding to a surface receptor can ultimately change the behavior of ion channels inside the cell, without those channels ever directly interacting with the neurotransmitter. The second messenger, CAMP in this case, acts as a relay signal that bridges the gap between receptor activation at the membrane surface and functional changes deeper in the cell. A second major intracellular signaling pathway involves receptors such as alpha-1 adrenergic receptors, muscarinic cholinergic receptors, and metabotropic glutamate receptors. When neurotransmitters bind to these receptors, they activate the enzyme phospholipase C. This enzyme cleaves a membrane lipid called phosphatidylinositol 4,5-bisphosphate into two distinct second messengers, 1,2-diacylglycerol and inositol 1,4,5-trisphosphate. Diacylglycerol remains in the membrane and activates a family of enzymes called protein kinase C. These kinases phosphorylate ion channels and can either enhance or suppress their function depending on the specific channel and context. Inositol 1,4,5-trisphosphate takes a different route. It travels to the endoplasmic reticulum and binds to a receptor on its surface, a receptor that is itself a calcium channel. When inositol 1,4,5-trisphosphate binds, this channel opens and releases calcium from intracellular stores into the cytoplasm. This calcium signal then activates a further array of calcium-sensitive enzymes, including phosphatases and kinases, which alter the phosphorylation state and function of additional ion channels and cell proteins. Together, these two pathways illustrate how a single neurotransmitter binding event at the cell surface can trigger a branching cascade of intracellular events, ultimately producing changes in multiple ion channels and proteins simultaneously, far more complexity than a simple ligand-gated ion channel could achieve on its own.